Hello, I'm Brian Malow, and I'm at the 70th annual Lindau Nobel Laureate Meeting, which, of course, unfortunately had to be virtual this year, but we do have more than 70 Nobel laureates participating and over 600 young scientists. And I'm speaking to one right now, Lorenz Adlung. Lorenz, thanks for speaking to me.
Pleasure is all mine.
And where are you? I'm in Lindau. Where are you right now?
I'm in Frankfurt in West Germany. I'm in a transition phase because I'm about to open my own lab, the Adlung lab, in Hamburg in the north.
You have jerseys for your own lab?
Yeah, I do. I used to customize this one for it.
Excellent. So where are you from? You are German, right? Where are you from?
Yeah, I was born and bred in Thuringia in the green heart of Germany. And then I went there to boarding school. And then I moved to Heidelberg in the southwest to study there and to do my PhD there. And then for a postdoc, I went as a visiting scientist to the Weizmann Institute in Israel for 3.5 years.
Did you happen to know of or work with Danny Shechtman, one of our Nobel laureates there?
Of course, I know him, but I didn't manage to meet him there. I met Ada Yonat.
Yes, I've spoken with her. Yes.
Yeah, she is also a wonderful person. So, but
yes.
Of course, they are both like idols to me.
Yes, and so tell me something about your research. What kind of scientist are you?
Well, I must say I don't like putting labels on people. So, if you ask a cell biologist, they would say, this is a mathematician or a computer scientist. But if you ask a mathematician, they would say, this is a molecular biologist or something. So I'm sort of a hybrid individual, and in the essence, it all comes down to methods that we are using to answer questions.
So what do you mean by that? Especially, I'm a non-scientist, and for the non-scientist, what do you mean it's about the methods you use, the methodology you said?
Yeah. We as human beings, we are composed of cells, right? And these cells do very well as we are just sitting here now, more or less healthy. But they also do worse if we are getting sick. And we want to scrutinize this and really understand how cells interact, how they communicate. And for this, we need technologies to measure individual cells and their molecules, their molecular makeup. This is something we use. The problem is not getting this data so much, because we also get this now from patients, but understanding it. It's so complex, and the more we know, the less we understand, or the little we realize that we understand. And for this, we also need computers and mathematical models to better make sense out of this data.
Yeah. So what sort of problems do you work on then with these methods?
Mostly now I'm focusing on metabolic research and the immune system. So I'm focusing pretty much on fat, adipose tissue, as we call it in science, because the fat secretes a lot of molecules that then interacts with cells from other organs like the gut or the liver or the kidney. And we try to understand this complex interaction, again, in health and disease.
Yeah, I'd like to actually hear a little more about that. Like some fascinating aspects about it. Do you have like specific questions that you most want the answer to?
So there's one sort of aspect that we are just about starting to understand, because, fat always, there is this, I don't know, like stereotype that obese people are always sick and it's not good to have excessive fat. But it's not necessarily true, because we just discovered a new immune cell type in the adipose tissue that actually protects you from metabolic diseases. So it's not always the case that the more fat you have, the worse it is. There's also some good fat, and we are just, again, on the cellular level trying to understand how this works.
That's so fascinating to focus on individual cells.
Maybe
probably
also
not smells, but individual cells, and even smaller units than that. And it's almost, I find when I contemplate the largest things like the scale of the universe, or the smallest things like insects, and then microscopic little like mites or something, and then getting down to the cell and the components of the cell, like, how does this work? How does how do these trillions of cells come together to make this body? And how could this relatively short piece of code that encodes for not really that many proteins, how does that result in me? I can't wrap my mind around it.
Yeah, and if you're talking about yourself, it's not just you. You know, you mentioned all these 30 trillion cells that you're composed of, but then there's unicellular organisms, in other words, bacteria that are inside your gut that to the same number, maybe even 10 times more, and they also make up yourself. You know, it's, we want to know something about ourselves, but we need to know some more about our cells and how they, you know, interact in these complex networks. It's really fascinating, and that's really what drives me. You know, this curiosity that I really want to figure out how it works.
And in recent years, there's been a lot of attention on the human microbiome and the role it has, and the fact that like you said, it's not even just our genes, because the genes in certain microbes inside us are doing things that, so their genetic code is important because of things they, so in particular, you mentioned obesity and fat. And I think that's an area that's been pretty exciting for some years, hasn't it? That so what role, because we tend to think that if someone's fat, it's because they eat too much, they don't move enough, that's it. But tell me something about the role of the microbiome that shows that it's a little more complex than that.
Yeah, you're absolutely right. It's not that simple. Just one example. So if you're saying it's just you're eating too much or the wrong things, but just assume there's two kind of bacteria, and one kind of bacteria can process all this food and sort of get rid of whatever is unhealthy about it or healthy about it, and the other one can't. So the fact whether you're having or not having one or the other bacteria makes you less or more susceptible to develop metabolic complications just given by the food that you're eating. So as you said, you know, it's not as simple, and we are really trying to now understand the big picture also with these technologies that now allow us to not only measure our own genetic makeup, but also the metagenome, as we call it. So hundreds and thousands of genomes that are actually inside us.
That's fascinating. And an example that I think a lot of people are familiar with is, like, we can't digest cellulose. And we know that cows can eat grass and digest cellulose, but the cow actually can't digest cellulose, can it?
Yeah, no, it's the microbes.
Yeah, that's so fascinating. So, and also just amplifying what you said, so two people could eat the exact same diet and one actually extracts more calories from that food. Is that true? That's what I've understood.
Yeah, there's a lot of variability between individuals, and this is also a big term right now that we try to, you know, tailor treatments to individual persons. That's what we call personalized medicine, you know, which is of course very expensive to develop a treatment for an individual person rather than for a big cohort of people. But we have the technology to do it, and sometimes, you know, to develop a personalized treatment, it only requires a computer simulation. So why not trying to do it now that we have all these methods and this data and this understanding at hand?
Yeah, and this, our microbiome. So again, in addition to just the food you eat and how much you eat and how much you move, there can be effects on the microbiome by drugs we take, and maybe what else? That's the only one I can really think of. But medications can affect our microbiome and then affect how much calories we get out of the same meal. But what's another thing besides medicines we would take that can have those effects?
I mean, antibiotics are obvious, right? But then also your physical activity, whether you're more active during day or during night, all this affects our physiology, but also the microbiome. And of course, they are all like interdependent. So it's really our entire lifestyle that affects our well-being and the well-being of the individual microbes that live inside us.
Can the time of day that I eat something, even me, the same subject, if I eat the same thing at night or in the morning or in the afternoon, do I potentially absorb different amounts of calories and other nutrients from it?
Absolutely, because again, as I said, the microbes are active during different times of the day, and also your gut is more or less leaky during different times of the day. So you can absorb more or less nutrients depending on when you're eating. And then, of course, it also depends on our roommates and neighbors, because, you know, we are leaving traces of ourselves also in the form of microbes everywhere, and people tend to pick them up. So
really, and
yes.
how is that addressed experimentally?
So this is basically, if we are looking at experiments, we usually have, or in one particular project that I'm working on right now, we have two different cohorts, let's say healthy or non-diseased and diseased. But then these two cohorts also differ, because in one we have just matched individuals that are not sort of connected, and in the other case we have different household members. And it's known that if people tend to share a kitchen or a bathroom, then at some point they're the bacteria that live inside them get more and more similar to one another.
Very interesting. And
Yeah.
have you another thing that's been a very interesting topic for several years, and I think going forward, fecal transplants. Do you have anything to say about that? Is that does your work ever address that? And could you tell us what fecal transplants are?
Yeah. So in essence, it's swallowing poop. Okay. We do this in our research, but only in mice. So a classical approach is you take, again, a certain cohort you're interested in, say a diseased and the non-diseased one. You take stool samples from both of these individuals, and then you transplant them to germ-free mice that don't have bacteria. And then some of the bacteria from the humans, they colonize in these germ-free mice. And maybe they get the same disease or not. And maybe you find some of these bacteria that are able to prevent the disease from coming up or getting as severe as it gets. And then, of course, the question is, if I transplant these bacteria or the stool they are living in, can we also use this to treat human individuals with this?
Fascinating. And what's your motivation for going into this field? First of all, did you always know you wanted to be a scientist? And if so, this type of scientist, what attracted you and inspires you to keep working on these sorts of problems?
Yeah, I think I was always a curious person, and it's really this curiosity that drives me. And I grew up in a very rural area on a farm, but I was reading these Donald Duck comic books, and there is Gary Gearloose. He's like an engineer, but he's an inventor. And I always thought, I had no science. I didn't actually know what a scientist is or what a scientist does, but I imagine, like, being in a lab, inventing things, finding new things, like every day, even though the guy, even Gary Gearloose, he fails like most of the time. I thought that would be amazing, but I had really no idea. But then I went to boarding school, which was like a school that specialized in mathematics and life sciences. And then I got a grasp on, okay, cool. Here I'm really, it's my job to find out new things, and it was amazing. I don't consider it work. I'm living the dream right now. So
yes, that's excellent. So I also wanted to talk to you about the meeting here. Unfortunately, it's virtual this year, but as we were leading up to the meeting, before the meeting began, what sort of anticipation did you have about it? It's a long process of actually, I've spoken to a couple academic partners, and I haven't spoken to any of the scientists about that process. So let's actually step back a minute. Could you tell me something about the process? I think a lot of young scientists that would like to attend Lindau would like to know, what was the process that you had to go through to become one of the attendees this year?
So first of all, I knew about the existence of this Lindau meeting for several years already. And I always dreamed of, becoming a part. And then when I was in Israel, I was lucky to win a Marie Curie scholarship from the European Union, from the Horizon 2020 program. And then the Weizmann Institute was allowed to nominate a few scientists to start this application process. And I was lucky enough to be selected. And then you have to write like a letter of motivation and send in your CV and certificates. And then again, I was lucky enough that I was selected once more, and then I made it to the meeting. But then also, unfortunately, the Corona crisis occurred and this pandemic. And last year there was only the Lindau Open Science Days. Which still I participated also in the hackathon, and it was a great experience.
Which project did you work on in the sciathon? Yeah.
We had now I have to rack my brain a bit, because it was also in a period when I was applying for positions. So I was in, and also I did a Corona hackathon.
Okay.
So I think we did. I don't remember the name. It was something with Compass and PsyCompass, exactly. So we wanted to bring together basically scientists as mentors from all over the globe to team up projects, particularly to give people from low-income countries a chance to, use also technology and data and expertise that is available in developed countries. We are very privileged not only to be in this meeting, but also to have access to all these technologies and infrastructure. And this is also something I like really about the meeting this year, that people are reaching out and asking for help. And here, of course, everyone is so supportive and offering advice, but also like hands-on, chat on the matter. So this is something I really, really enjoy.
Okay, I want to come back to that in just a moment, but tell me, so in the lead up to the meeting, tell me about some of your anticipation. What were you looking forward to?
Yeah, so of course, first of all, it's about meeting all these, meeting your idols, people who won a Nobel Prize. They can be very inspirational. And I was lucky enough to already meet some of them because whenever there was, a lecture somewhere around in Germany, I was just traveling to see these people talk and actually reach out to them. Because
do you want to
it's
mention
really,
a few in particular?
For instance, I met Jim Allison already, who's going to still have a session tonight. I'm really looking forward to, because it's always great seeing him like talk. But also before they got the Nobel Prize, I was able to meet Jennifer Doudna and Emmanuel Charpentier, which was a great experience. And since I did my PhD at the German Cancer Research Center in Heidelberg, of course, I also was lucky to meet Harald Zuhausen and also Stefan Hell. And these, as I said, this is really invaluable. I mean, priceless to exchange with these kind of people, but also to meet other people who want to see them and see their reasons to come and how they interact. We are sometimes a bit, shy and humble, but they are the Nobel Prize laureates. They are humble themselves, and it's just really amazing to see all these different individuals. Also, it's not that they have all the same personality. Some are really outgoing, and then, for instance, Ada Jonat, she's a bit more not hesitant, but she prefers to have like private conversations. I feel like,
Yes,
which I totally accept. That's.
I know. I interviewed her the first year I was here in 2016, and I really enjoyed interviewing her. And the video is available. I know it's on the Lindau Facebook, on the Lindau YouTube channel, and it's probably somewhere on the Facebook page, but it was a few years back, and Facebook's not great for seeing old videos and old posts, but I enjoyed speaking with her, and I asked her, why does she keep coming to the meeting? Because she's a laureate who has come year after year. She keeps coming. So I said, why do you keep coming to the meetings? And she said, I come for many reasons. Being interviewed is not one of them.
Yeah, it fits.
Yeah,
really fits.
and it really is. They're not coming for their own glory or anything. They are there for the young scientists. So that's fantastic. And you know what? So I haven't had a chance to ask anyone about this. So many things about this year's meeting are completely new and unique, untried. And so we have these networks, so it's virtual, and we have these two networking platforms of Hopin and Wonder. And you already touched on this. I want to hear from you how that's working for you. I want to hear it from, because all I know is from this side of it. But as a young attendee, you mentioned it already. So let me please elaborate on that. Like, how are those networking programs working for you? Like you said, people are reaching out and connecting.
Yeah, especially during these breakout sessions that are usually in the evening. So yesterday I had the chance to finally see also Elizabeth Blackburn, whose career I was following intensively. And yesterday she was there, and she's also such a lovely character. And in these formats, actually what's unique, I think it's not only that individuals can talk to them one-on-one, but also they really take the time. It's not like, okay, one question, you have half a minute and then we have to move on to the next, but really it's young scientists who have also a couple of questions and follow-ups, and they really take the time to patiently answer them. And what I really like is that they share their enthusiasm. I mean, that's a big motivation for me. I'm getting very enthusiastic about science and talking to scientists, but also talking to the lay public about science, and seeing that this resonates, empathy, after all, that people feel excited about science,
Yes.
even if it means failing most of the time. But they keep trying and they keep asking these questions. They stay curious. That's something that I really take from this meeting and from this format in particular, that you have the time to do this one-on-one chats.
Yeah. So you mentioned some laureates, but if you were here in person, you so easily connect with so many people, so many of the other attendees. Have you been connecting with other young scientists this year?
I'm trying. I'm still in the process, because also I'm starting my own lab now, so I'm basically recruiting, right? So all these people, they would be perfect candidates, and then talking to people in person, you aside from the scientific aspects, of course, that need to match, you can easily feel whether you connect, because I think this is also essential for a good lab environment and
Yes.
An atmosphere that I want to cultivate. It we are positive, we are there as scientists, as individuals, but also we are a team, and we should share common values that are also now in this Lindau guidelines, which I really appreciate, and which I will also incorporate for my own lab to a certain extent, that we can also connect, on a personal level. And I'm trying this now. At the moment, I have more the focus still on the laureates, because there's still so much more I want to know from them. But at the same time, I'm trying to connect also to other people as well as I could.
Excellent. And you mentioned your interest in outreach. Let's talk about that, because you have quite an interest in outreach and you've done a lot of different things. I don't know where to start, but why don't I just ask you what a science slam is? I understand you've participated in a lot of science slams and poetry slams. Tell me about that as scientific outreach.
Yeah, it's a form of science communication where basically you go on stage, you talk to the lay public, and you have like 10 minutes to tell in an understandable way, even for lay people, what your research is about and what you're doing. And it should be informative, but it should also be entertaining. So
what sort of venue would that take place in? It's not a lecture hall, I'm guessing.
No, it's you can do it at a pub, at the bar or something, as long as you have some, a crate of beer to step on to get some interest. But also you have like all cinemas. I've been performing all cinemas and theater houses, opera houses, wherever you have a stage, basically. Sometimes people do it with a presentation and depending, but then sometimes it's also just, spoken word, poetry slam is more like just spoken word, not necessarily on science. But I also wrote poems and songs on my scientific research, for instance. I did this also in Israel. And it's just a wonderful experience, because if you give a talk at the scientific conference and you do very well, people might applaud. But if you do a science slam and people connect, to your enthusiasm, and I always do this rap thingy. So once I start to rap and the beat kicks in, people start to cheer and bounce. And this is priceless. Again, a really unique experience that I think I would never get at a scientific conference. But maybe that's a challenge, that now organizers want.
That's what I was going to ask you. Have you ever considered doing that sort of delivery and that sort of presentation with the rapping or the poetry at a scientific conference where they're expecting a very straight presentation?
I was booked for science fairs yet, which was as close as I can get. I wasn't asked for a scientific conference. I'm going to one in Berlin next week. I might ask the organizers what they have in mind for the evening program.
So I guess do you love the process of scientific research, the actual research you do, and is that something you love? And what do you love about that? And then I'm going to ask you, in comparison, what drives your interest in outreach? Why is that important to you, and what do you enjoy about it?
Yeah, so the question that drives me is as simple as the following one. It's why. I mean, why are we healthy right now? Why are we getting sick as we age or as we eat the wrong food? I just want to understand how this happens. As I said, I'm very curious, and now we have all these methods, and I was very lucky to receive good training, have wonderful mentors to teach me all these tools that I need now to answer these questions to a certain extent. We won't ever know everything, but we have to connect, small dots, and maybe tomorrow we find out something new, and what we found out yesterday seems to be all wrong, but that's, the scientific process. So that's what drives me. I want to know how it all works, how all our cells are connected and stuff.
And then on the outreach part, could you tell me, is there anything similar about those two things, or how are they different, that these two different sides of things? Or is there something that connects them that you think these actually two different seeming things, your research and your outreach, is there something that connects them,
Yeah. So if the science is on the one side and the public is on the other side, I mean, they also want to know, and they have a right to know because they are funding our research, right? And so it's also our obligation, I believe, to tell them what we are doing in simple terms that they are able to understand. It's not that they are, not intellectually capable of doing it, but they are just coming from a different area of expertise. Maybe they are caretakers, they're experts in fields that I could never work on because I don't have this expertise. But now it's my obligation, and I also want to do that. I want to share my enthusiasm. I want to share my open questions, because this is, first of all, a precondition that they accept science. Nowadays it's all about fake news and stuff, but now in this pandemic, we are about to solve it thanks to science. This vaccination campaign, all these prevention measures, they are based on a solid scientific foundation. And if we share our concerns, all this uncertainty that we don't know, we also should be open about uncertainty and failures and all the stuff we are trying, which doesn't work eventually. This creates acceptance, but then if we share our enthusiasm, it will also create more sympathy for science, and people will look more positively on science and the scientific endeavor as such, which is important to us scientists, but also to us as members of the society.
Excellent. Thank you so much. I know you're in the midst of this meeting and I don't want to keep you from it any longer, but I know you have a website and it gathers together a lot about your research as well as all these many outreach activities you've done. So you want to tell people where they can find you on your website and on social media, perhaps?
Yeah, so my website is just my name, lorenzadlung.com. And that's also my handle for Twitter and Instagram and all these other networks. So it will be pretty easy to find me there. Also on YouTube, if they want to see some of these raps, I have them in German and in English, and Science Slams in German and in English. So,
and you did a TEDx talk at Heidelberg.
Right. It was also about how interdisciplinary science can. It was entitled, "Let's Talk Science. It Saves Lives," and this is my firm belief, really. So we should keep going and watch other Nobel Prize laureates talking science, because this is what we're here for.
Excellent. Well, thank you so much for talking to me, and enjoy the rest of the meeting. And then I think, and as I like to keep saying, educate, inspire, connect. And these connections, you'll find in the coming years that you will probably have friendships and possible collaborations out of all this. So yeah, thank you so much, and enjoy the rest of the meeting.
Thank you very much indeed.